Hey everybody, thanks for joining me today. Remember that this is a lecture based on EKGs and actually electrophysiology. And we're going to start from the atoms all the way out to the ECG itself. So we need to build this base concept, and these base concepts are gonna be our foundation from which we are going to grow and begin to understand and unravel this mystery of electrophysiology, because it's really easy to just look at an ECG or an EKG or whatever and just correlate it to something. But it's more important that you build a foundation that you can work upon to further analyze, because if you just get into the habit of saying, oh, ST segment elevation is here because it says so in the book, that's sort of a lazy way of thinking. So first and foremost, let's begin with some base concepts. Now, these base concepts always have to start at the atom level. So these atoms I picked for a particular reason, because they're going to be utilized later in the lecture. But remember that these are our base atoms when we talk about, the sharing of charge, and specifically the ones at the top: sodium, potassium, calcium, chloride, and then oxygen and carbon. You can pretty much tell that, those are going to be utilized as the basis of life. We have organic chemistry that's based on just those molecules alone. But let's go into why they're important. Because when these atoms sort of collide together, they make in our body a special molecule, and there are a couple special molecules called purines and pyrimidines, and you don't need to remember the names of these. Just understand that they coincide with other molecules like pentose and sugar and oxygen to make DNA. So these molecules that are made out of atoms make DNA, and then this DNA is sort of lined up along this pair, along this phosphate backbone, and it's wrapped around these histones, right? These little proteins, they're wrapped around these, and you can actually see how intricate that is and how crazy that is. And they're jammed inside of this chromosome like spaghetti, just wrapped around miles and miles. You could think of it, miles and billions of miles around, into this very, very small, tight-packed compartment that's then placed into our nucleus. And our nucleus houses all this information. The way I like to think about it is that DNA is like code. It's literally like the code on your computer. It codes for certain aspects or programs to be run at a certain time. And these codes can differentiate in small ways and make a monumental difference in the programming. So if you think about, like, if you were to get one key wrong in HTML, it really fucks the whole program up, and you can't work with it. So this is a really cool way to look at it. So I like imagining that it's code over here that's actually causing that. So let's see what we're coding for right now. So as you can see, the pericardium, the epicardium, the myocardium, and the endocardium. These words should be, sort of clicking something in your head saying, oh, I know that's the heart, right? Cardium is the heart, and it's separated into these 4 layers. Some say just 3, and they count this as 1, and rightfully so. We can see that they're actually laying on top of each other, but they are a little bit different. And these visceral layer, the way you can remember it, epicardium is the visceral layer, meaning it's attaching to the organ itself. Then the myocardium, and you can actually see the difference in the structure of these cells that this coding is having. You can see the major difference in these cell types. These intercalated discs right here, and how they're going to contract later on. And then we have the endocardium, which actually not only covers the inside of our of the heart, but also the leaflets. It's very, very cool how it just sort of folds into it. But the myocardium is where we want to focus today, because the myocardium actually has 2 different types of cells. They have pacemaker cells and contractile cells. So the autoarrhythmic, it's really easy to sort of intuitively say, oh, autoarrhythmic. It makes its own rhythm, and then the myocytes are the contractile cells that actually contract and pump blood. But as you can see, the DNA, the code right here, they're really alike. They're almost identical to what they are, or excuse me, to each other. But the important fact on how they differentiate is within inside the cell membrane. So we see this normal cell right here, and yes, I did spend about 30 minutes putting, correlating the stickers to the different types of organelles within inside a cell, but we are actually going to take a dive into the membrane. So we see this bilipid layer right here, right? And then we see these channels and these little proteins right here and these pumps. And there are actually a lot more. This is just a simple representation. But what these channels and pumps do is they move atoms in and outside of the cell. And when these atoms move inside and outside of the cell, it creates a charge. It creates a difference in potential, an electrochemical potential in between the inside of the cell and the outside of the cell. So that's how they change. That's where this differentiation comes into place, because the pacemaker cells actually have more of these channels. And when we have more of these channels, it's easier to create a charge differential. It's easier to create a difference in charge from the inside of the cell to the outside of the cell. So that's why it's so important to understand that the difference between these two are mainly boils down to the amount of proteins and channels within inside the cell wall. Because the reason that this is important is these ions carry charge. That's how they do it. Normally when these atoms are inside of a solution within inside of our body, within inside of our blood, they ionize, and we can see potassium over here becomes a positive ion because this electron goes elsewhere. We can see the sodium and calcium, how they all have positive ions because electrons are moving outward. And chloride is one of the only ones that has a negative charge to it, solely because of its electronegativity and it wanting to have electrons to it. But that's the thing. If we think back to that bilipid layer, it's the movement of these charges, these positive and negative charges inside and outside the membrane, that cause automaticity. And that's what this whole first video is about. So this is the easiest way I can sort of break it down. Automaticity is the amount of available channels or proteins that allow charge to flow through to create an electrochemical gradient. And that difference in potential is what causes hearts to fire. Because we can't really think of, these cells as actual neurons. They're not really neurons. You can't think of these autoarrhythmic cells as the neurons that are in your leg and your hands. They have a completely different world to them, and their different world is mandated and controlled by those different channels and those different proteins that are on the surface of the cell that makes them more applicable to fire and become autoarrhythmic. And that's what automaticity is. And that whole question of automaticity was just baffling to me, that I couldn't tell you, how this works. How do we fire? How do our cells, start to fire? How does the SA node to fire? How does the SA node to fire first? What are the differences between the SA node and the AV node? And it all boils down to the amount of proteins and channels to allow these ions to create an electrochemical gradient. So that's the basis of everything. All right, great job, everybody. Thanks for following along. We'll see you on the next video.